My name is Tiffany Callahan, and I'm a postdoctoral research fellow at Columbia University in the Department of Biomedical Informatics. My poster is on a project that was conducted as part of my dissertation, which examined the utility of aligning a framework for standardizing data quality checks, originally designed for electronic health record data, to ontologies like those in the Open Biomedical Ontology Foundry. Ontologies play an important role in standardizing and integrating knowledge and are subject to data quality issues. Quality assessment is vital to ontology development and maintenance, and is typically performed with the goal of identifying logical or semantic errors, lexical or syntactic errors, and structural or hierarchical errors that are evaluated at both the schema and data level. While many existing ontology quality assessment tools exist, they can be difficult to use, are often inconsistently maintained, and may not even be publicly available. And in fact, most of these tools evaluate only a single ontology. And there are many challenges to merging or combining multiple ontologies, and while solutions to these challenges are currently under development, there are no existing tools to robustly evaluate this. Within the clinical domain, many clinical data quality assessment strategies exist, and there was very little consensus on what mattered most and how to best assess it. The Harmonized Data Quality Framework, or HDQF, was developed by Michael Kahn in order to address this problem. The figure in the upper right-hand corner of the poster provides a high-level overview of this framework. And at a high level, this framework describes a set of categories that operate within two data quality assessment contexts. The first one is the confirmation of expectations about aspects of the data that are based on comparisons to local knowledge or pre-specified metadata. And we call this verification. The second is comparisons to external benchmarks and gold standards, and we call it validation. Within these contexts, there are 3 dimensions and further categories within each dimension. The 3 dimensions include conformance, or the assessment of whether or not data values adhere to pre-specified internal or external formatting constraints like value, agree with structural constraints like primary and foreign key relationships like relational, and are accurate when computationally derived from existing variables like computational. Completeness assesses whether data values are present at a single point in time, which is atemporal, or multiple time points, which is temporal. Plausibility assesses whether data values are believable and is assessed through the agreement of logically constrained measures or distributions and independent measurements of the same fact, temporal properties or sequences, and state transitions of measures, and through the presence of duplicated measurements, variable values, or records. The goal of the work presented in this poster was twofold. We aim to develop and evaluate ontology checks to assess the fitness of use of individual ontologies and merged biomedical ontologies, and we wanted to characterize whether these checks could be mapped to the HDQF. For ontology check development, checks were reviewed by a member of the Open Biomedical Ontology Foundry working group and a semantic web developer with over 10 years of experience. These checks were applied to the 11 biomedical ontologies, which are listed on the poster. For check characterization, the developed ontology checks were aligned to the HDQF under 2 contexts, a single ontology or as a set of merged ontologies. The HDQF developers then independently mapped the ontology checks, and any check not able to be clearly mapped was discussed until a final mapping consensus was reached. In total, we developed 7 data quality checks, which are listed in Table 1. And the checks included the assessment of deprecated and obsolete entities, verification of value errors like typing, examination of semantic heterogeneity, alignment of identifiers and identifier errors, and search for punning errors. All checks that we created, except deprecated and obsolete entities, were mapped to an HDQF category, and this led to the development of a new data dependency category. At the individual ontology level, 42.8% of the ontology checks were mapped to the value conformance category of the HDQF, 28.6% were mapped to plausibility uniqueness, and 28.6% were mapped to the new data quality assessment category of the HDQF. And at the merged level, all of the ontology checks mapped to relational conformance. With respect to categorizing the checks, the cell line ontology had the most errors, and the vaccine ontology had the fewest. The amount of deprecated entities ranged widely across the ontologies, with KEBI, the sequence ontology, the gene ontology containing the most. Punning errors were identified in the cell line ontology and when working with the merged set of ontologies. and semantic heterogeneity errors were only identified when working with the merged ontologies. So in total, we developed 14 ontology checks and mapped them to the HDQF. We extended the HDQF by adding a new data dependency concept. And while not all HDQF concepts were able to be aligned to a new ontology check, this provides opportunities to help guide us into future quality check development. These results then provide initial support of the HDQF as a translational tool for standardizing data quality checks, but there are several important limitations, which include ontology checks were only tested on a small sample of ontologies. The quality pipeline does not take advantage of existing tools like Robot, but it should. And ontology developers did not actively participate in the HDQF alignment process. These are all limitations which we aim to address in future iterations of our pipeline. And we'd like to acknowledge that this work would not have been possible without the co-authors listed: Bill Baumgartner, Nico Matsunoglu, Nicole Vasilevsky, Larry Hunter, and Michael Kahn. And we'd like to thank you for listening to this recording and for attending ISMB. Thank you so much.